Hello, this is Dr. Subhradeep Karmakar. Welcome to PG Pathshala. I am an assistant professor in the Department of Biochemistry at All India Institute of Medical Sciences, New Delhi. So, today we are going to talk about C-value paradox and the DNA complexity. So, the genome size is defined as the DNA length of one haploid set of chromosomes. It varies greatly between prokaryotes and eukaryotes, as well as within each of these organisms, two organisms. So, within the prokaryote the genome size is different, between the eukaryote the genome size is different. Number of proteins which is encoded by the genome also increases with the increase in the organism's complexity, in addition to the tissue specific encoded proteins. Hence, the genome size can be roughly correlated linearly with the DNA complexity. It is due to this reason that prokaryotes has a smaller genome, because the complexity is less obviously as compared to the eukaryotes which has a much larger genome. However, with the advent of next generation sequencing techniques like NGS and more genomes has been sequenced because the cost has been reduced, it becomes apparent that the gene content does not exactly correlate with the genome size. And this essentially gave rise to today's topic, the C-value paradox. The fact that the different organisms have the same general level of biochemical, organic and morphological complexity often have widely different C-values. For example, among genomes of vertebrates, the C-value ranges from the DNA content ranges from 0.5 to 150 picograms. So, this is very weird that the organism's complexity does not correlate exactly with the content of the DNA. So, introductions for today is that C-value enigma and see what does it stands for. It is also expected that more and more complex the organism is, the more amount should be of the DNA. It should have and therefore it should have a very linear relationship. However, it is found that for a slight increase in the number of the genome, the genome size actually increases numerous or many folds. And the C-value paradox is basically this, that how can we account for the amount of the DNA in terms of the known functions. Now, very similar organisms can show a large difference in the C-values. Now, the amount of the genomic DNA in complex eukaryotes, for example, is much greater than the amount needed to encode the proteins. For example, mammals have 30,000 to about roughly 50,000 genes, but the genome size is about 3 billion base pairs, right? In the genome sequence comparisons in organisms with bigger genomes across a broad range of species revealed that a universal general pattern that organisms with bigger genomes tend to have more genes and more number of introns than organisms with smaller genomes. And the smaller genome is sometimes we do not have even the introns. This is particularly apparent when multicellular eukaryotes are compared with unicellular prokaryotes, or when slow growing animals like the humans actually when compared with fast growing animals like Drosophila. In recent paper, Michael Lynch and John Corney had proposed a general model to explain these broad patterns. Their idea is that the differences in effective population size, NE if you say that, and the differences in all three features of the genomes. NE provides a general measure of the rate of the random drift, the process of random fluctuations in the allele frequencies which is inevitable in finite populations. In an idealized population of N diploid, randomly mating adults with equal expected reproductive successes, selectively neutral variability has been lost at a rate of 1 by 2 to the power of 1 upon 2 by n per generations. And in addition to this, the species may be actually divided in partially isolated subpopulations. In cases the rate of the drift is equal to 1 by 2 NE. Usually NE for the species as a whole is much smaller than the actual number of breeding individuals. So, what are the objectives for today's class? The DNA as a genetic material, number one. Number two is that functional output of the DNA. Number three is the transcription of the gene expression. And the number four is what is C-value paradox and DNA complexities. And the number five is implications in the outreach of the everything, and also how the modern biology has got to know a lot about the C-values. The DNA as a genetic material was proposed by Frederick Griffith in 1928 by carrying out a similar experiment with Streptococcus pneumonia, bacteria that causes pneumonia. He observed that when these bacteria, that is Streptococcus pneumonia, actually being grown on a culture plate, some of them produces what he calls as smooth colonies, shiny colonies, S colonies, whereas the other produces rough colonies or R colonies. And the differences in the character, that is the smooth versus rough, is due to a mucus, which is mucopolysaccharide coat, which is present in the S stain and not been present in the R stain. So what experiment Griffith did is that he injected both the S and the R strains into the mice, and the one which was infected with the S strain developed pneumonia and died, while the one that is infected with the R's or the rough strain stayed alive. In the second stage, Griffith killed the S stage strain bacteria and injected them into the mice, but the mice stayed alive. Then he mixed the heat killed S and the live R strains, and this mixture was injected into the mice and they also died. In addition, he found that the living S bacteria or S strain bacteria is dead in the dead mice. So from the conclusion, based on this observation, Griffith concluded that the R strain bacteria has been transformed by the S strain bacteria, and the R strain inherited some transforming principle from the heat killed S strain bacteria which made them virulent. And he assumed that this transforming material is actually the genetic material which actually get passed from one bacteria to another one. Now the functional output of the genome. What do we know about that? Now the genetic material is actually been stored, we know, in the form of DNA or deoxyribonucleic acids in most of the organisms. In humans, the nucleus of each cell actually contains roughly about 3 into 10 to the power 9 base pairs of the DNA, which is distributed about 23 pairs of chromosome, and each cell has 2 copies of the genetic material. This is known collectively as the human genome. The human genome contains about 30,000 to 40,000 genes, and each of which actually codes for 1 proteins. Now the process of transcription actually begins when an enzyme that is known as RNA polymerase or RNA pol actually attaches to the template DNA strands and actually starts catalyzing nucleotides producing a strand of the RNA. Polymerases are very large enzymes composed of dozens of different subunits, and when active on the DNA, they are also typically complexed with other factors known as the transcriptional complex. In many cases, these factors signal which gene to be transcribed. Now, the transcription is the first step in the gene expression, in which the information from a gene is actually being used to construct a functional product such as a protein. Now, the goal of transcription is to make the RNA copy of the gene's DNA sequence. For a protein coding gene, the RNA copy of the transcript carries the information needed to build a polypeptide or a protein subunits. Eukaryote transcripts needs to go through some kind of what you call as a processing or QCs before it can be translated into proteins. The 3 different types of RNA polymerase exist in the eukaryotic cells, whereas bacteria has just only one. In eukaryotes, RNA polymerase 1 transcribes the gene that encodes for most of the ribosomal RNA or rRNA. RNA polymerase 3 transcribes the genes for small RNAs plus transfer RNA that plays a very important role in the translational process as well as other regulatory RNA molecules. Thus, it is the RNA polymerase 2 that transcribes the messenger RNA, which serves as a template for production of the protein molecules carrying the information from the DNA to RNA. Transcription of the gene expression. Now, the transcription is the process by which DNA sequence is actually being copied into a complementary RNA sequence by RNA polymerase. Now, the free nucleotides exist in the cell as nucleotide triphosphates or DNTPs or NTPs, which lines up opposite to the complementary bases. We know that A pairs with T, C pairs with G. RNA polymerase covalently binds the NTPs together in a reaction that involves the release of 2 additional phosphates. Now, the 5-phosphate is then linked to the 3 end of the growing mRNA molecules, and hence the transcription occurs in 5' to 3' direction. Now, let us have an overview of the transcription process. The process of the transcription can be divided mostly into 3 steps: initiations, elongations and terminations. What happens in initiation is that the RNA polymerase binds to the promoter and causes the unwinding or opening and separating out of the DNA strands. We call the DNA melting, right? And elongation actually occurs as the RNA polymerase moves along the coding sequence, synthesizing RNA in 5' to 3' directions. Now, when RNA polymerase reaches the terminator or the end, then both the enzymes and the nascent RNA strands actually detaches from the DNA templates and unwinds. Now, many RNA polymerase enzymes can transcribe a DNA sequence sequentially, producing a large number of transcripts. Now, in eukaryotes, there is something called the post-transcriptional modification of the RNA sequences necessary to form mature mRNA. Now, genome organizations. Now, let us have a synopsis of that. Now, what do you talk about the Cvalue paradox, different classes of the DNA, repetitive DNA diseases, and if a protein coding portion of the human genome makes up 1.5%, what is the rest of the DNA been doing? The transcription of the RNA, tRNA by RNA polymerase 1 and 3. Other coding RNAs and small nuclear RNAs, micro RNAs, non-coding RNAs are there. Now, let us have a definition of the genome. The genome is actually the total amount of the genetic material which is actually stored in the genome, and this is referred to as the DNA content of that particular cell. And in humans, we know there are 46 total chromosomes, which is distributed into 23 pairs, right. It is the nuclear genome that actually defines the multicellular organisms. It will be same for almost all the cells of the organisms. You can have organelle genomes as well, as like mitochondria has its own genome, like mitochondrial DNA. When you want to identify or distinguish one organism from another, such as in the forensic testing, you investigate the genome. So, genome actually gives the blueprint of that particular individual, very specific for that particular individual. Now, the transcriptome, the total amount of genetic information has been transcribed in the form of an RNA, is known as transcriptome. Now, this information will be stored as RNA. Now, the transcriptome is unique to the cell types and is being measured by the gene expression. Now, different cells within an organism will have different transcriptomes. Cell types can be actually identified by the transcriptomes. So, the transcriptomes is some kind of a blueprint for a cell. The cell is actually being the complete protein output. This reflects all the mRNA sequences translated by the cell. Cell types has different proteome and thus can be used to identify a particular cell. Genome investigations. What can you learn? What are the number of the investigations? There are a large number of investigations that can actually be carried out on the genomes of the different organisms, and this is known as comparative genomics and is an extremely emerging promising field of study in its own right. Having access to the sequence of a large number of genomes has opened up a plethora of opportunities. Even before the complete sequences actually were available, a large number of parameters could be measured in various genomes, and some informations about the genomic organizations, its architectures, its information contained in the multicellular organisms could be obtained. Some of these measurements has actually been made. Number one is that the base composition of the vast array of organisms, both prokaryotic and eukaryotic. Number two is that we know about the repetitive sequences, unique sequences, and the sequence content of a DNA, and how exactly is the sequences different from the different region of the DNA. The genome organizations, the base compositions of the DNA is a fixed value and is actually expressed as percentage of G and C of the genome. The variation of this value between the different prokaryotes is large, but this is surprising given the fact that different proteins actually produced by the species have similar amino acid sequences. The prokaryotic genome DNA can have as little as about 25% G plus C in Mycoplasma genitalium to high as much as 72% in Micrococcus. Eukaryotic genome DNA does not display the same variation between the species. The percentage of the G plus C compositions of most of the plants and the animal species falls in a pretty narrow range, averaging about 39 to 40%, with a variation about 6 to 7%. Base composition within the genomes. In prokaryotes, the bases are distributed evenly throughout the genomes with a slightly lower G plus C content in the promoters and the intergenic regions. This often has an 8 plus T segments which melts more readily than G plus C regions. Now, the relatively constant base distribution within a given DNA genome or bacterial genome actually suggests that although there may be unequal nucleotide pool sizes inside the cells, the system will have evolved many over generations to be like, and the rate of the DNA replication is constant. Now, the distributions of the G plus C content throughout each genome is in eukaryotes, however, vary significantly unless prokaryotes, whereas the mean variation of percentage of G plus C content within the eukaryotic E. coli genome is only 8.6. In eukaryotes, this variation is over 30%. Certain regions of the eukaryotic genome DNA are found to have A plus T rich, whereas certain regions are found to be G plus C rich, and this varies actually depending upon the different locations of the DNA. Now, the genome organizations, repetitive regions, unique regions, and the C value is the total number of the DNA bases in the genome per haploid cells of the chromosomes. When you compare this to the complexity of the organisms, you find a massive disparity. Some of the organisms seems to have far too much amount of the DNA for the complexities what they have. For example, the carp, the fish, has about 52 chromosomes, where the alligator that eats the carp has 16. Some flowers has far more genetic material than that of the DNA. Clearly, the amount of the DNA is not proportional linearly to the amount required to produce all the proteins made by the organisms or the position in the food pyramid or the food chain. Like example of the organisms, their genome sizes, number of the genes and percentage of the copy number, you would see in the literature that there is a disparity between these two. Now, say for instance, we see that this linear correlation is even further been deviated in the eukaryotic genomes. In prokaryotic is still okay, but in the eukaryotes we know that there is a huge deviation between the complexity of the organisms and the DNA content of it. Okay, going back to the melting and the reannealing of the DNA, which is actually been covered last year, interesting technique was actually been pioneered about 20 years back, and that is what is known as the Cot plots of the DNA. But it is the first hint that the genomic sequence is actually complex and it doesn't correlate linearly. Now, what is the C-value paradox or the Cot value paradox? Now, the DNA from a species and take it in, cut it into small pieces, and then you can actually melt it. Now, once it is in the form of single stranded, we can actually measure the absorbance 260 nanometers. Now, if all of the sequences actually been represented just once in the genome, then the reassociation curve or the reannealing curve of the DNA will follow a particular kinetics. But it is actually found that when you take a DNA of a complex organisms like a human and then actually melt it and then actually reanneal it, then there are certain regions which actually anneals very faster, there are certain regions which actually anneals very slower. So, there is a differential rates of the annealing of the DNA. Now, those sequences that are the most repeats will going to hybridize quickest, and those with the multiple copies also is going to hybridize quickly, while the more complex single copy sequences take the longest to anneal. Plot of the curve with absorption of 260 on the y-axis and basically time is going to give us what you call as the C plots. It is second order kinetics because it is bimolecular reactions, and the rate of the association is actually proportional to the concentration of both the strands. Now, the rate of the rehybridizations is dependent on the complexity of the DNA. Now, the complexity is actually been defined as the number of the bases in a unique sequence. For example, poly U has complexity equal to 1, and the sequence, suppose AGTTCA GTTCA, has about a complexity of 5 or 6. Now, the Cot value for a given sequence of the DNA is dependent on its complexities. Now, the plot of a complex mixture of the DNA sequences has a biotriphasic look about it, right. Essentially, the human genome DNA Cot curve has actually 40% of the fast annealing low Cot sequence elements and 60% of the slow annealing high Cot unique sequences, that is 1 copy per genome. The human genome can be divided up to into 4 classes: highly repetitive sequences which are present hundreds and millions of copies, moderately repetitive sequences contains about tens to hundreds of the copies, slightly repetitive sequences about 1 to 10 copies, and just single copy sequences, just 1 copy per genome. Now, the repetitive DNA by sequence analysis was found to contain more short repeat sections found in the satellite DNA. Highly repetitive sequences, one group of the highly repetitive DNA is the simple sequence DNA, which contains thousands of copies of a simple sequence which is actually repeated tandems numerous times. Now, the repeat sequence can be as short as 5 bases. Now, the repeat sequence are known as the short tandem repeats or STRs. The microsatellites, what are these? Okay, microsatellites are about the repeats, and mini satellites are again of the repeats or the different sizes or different number of repeats. These sequences are often been found clustered around the centromeres and the telomeric regions, and their transcription inactive. Now, the term satellite came from about the behavior in cesium chloride gradient, because many of these simple sequence repeat DNA is AT rich, it has lower density than more GC rich genomic DNA, and therefore is branded differently in gradient form a satellite. This repeats accounts about 10 to 20% of the eukaryotic DNA. Now, the moderately repeated DNA includes those sequences which has multiple copies in the genome, designed to increase the rate of the amount of the gene product, and some regulatory sequences which is actually been found scattered throughout the genome. Sequences such as the ribosomal RNA and the transfer RNAs, which are actually been required in large numbers, forms protein synthesis, has many copies in the genome. Now, the histone sequence also have large copy numbers in the human genomes. This means there are so many copies of the histone genes, and the group of moderately repetitive DNA sequences are those that scattered throughout the genome, known as the sign elements. Also stands for short interspaced elements, and lines elements stands for long interspaced elements. There are about 300 base pair longs, and about 1 million of such sequences exist scattered through the human genome. They account for about 10% of the human genome. They are actually been transcribed into RNA, but they have no known functions. They are known as ALU elements or ALU elements because they contain the regular recognition sequence of the restriction enzyme ALU. Now, the most common line elements in the human genome is the L1 element, a 6000 base pair fragment which actually has been repeated something like 50 to 60,000 times in the human genome. Now, the L1 sequences have also been transcribed on some in codes for some proteins, of course very less. Their function in the cell is actually unknown, Both the ALU and the L1 sequences are transposable elements capable of moving to the different regions of the genome, and therefore they could be potentially dangerous. Now, the group of sequences with a small number of copies on the genome includes such sequences such as the globins. This family of genes actually contains a number of closely related sequences varying only by a few bases in the coding, and is going to cross hybridize if we are going to do an hybridization experiments. This also known as the gene clusters. Now, the final group is the single copy sequence, makes up a vast majority of the genes of the human genome, very critical genes, just one copy, the gene being functional unit which codes for a single polypeptide chain. Now, this group is the most complex and takes the longest to reanneal because there is just one copy, so they do not have enough information to rehybridize back, and hence the log scale on the time. The highly repetitive DNA reanneals faster, while the complex single copy takes hours to even days sometimes to reanneal. So, our genome actually contains highly repetitive DNA which does not code so many proteins, and also it contains some multi-gene families and multi copies of the same genes. This makes about 40% of the human genome by cut analysis. What of the other 60% unique sequences? Only 1 to 2% of the human genome is actually coding sequence, and this poses a discovery or a discrepancy. Further investigations of the eukaryotic genes, that is 60%, that has been found to be interrupted by large sections of non-coding regions called introns. Now, this does not happen in bacteria. Bacteria does not have any introns. Even in the eukaryotic, there are some genes which does not have an intron, like the histone genes. Now, the coding sections are actually called the exons. Of course, you know that the exons, but don't confuse the exons, always protein coding, sometimes they also code for the untranslated regions, UTRs. Pseudogenes. The eukaryotic genome also contains pseudogenes that actually occupy a significant portion of the genome. Now, there are actually two classes of pseudogenes. One is known as the class 1 pseudogenes that has actually been arisen by gene duplications, and then they have actually been subsequently inactivated by various mutations. That is known as by insertions, substitutions, etc. Now, these pseudogenes actually been formed are often been found near the functional gene counterpart. Now, the second class is known as the type 2 pseudogenes, actually been processed sequences which lacks introns and often contains a vestigial poly A tails, and has actually been originated during the evolutions from mRNA that has actually been copied by a reverse transcriptase enzyme back into the DNA. Now, the sequence that was then been inserted back into the genome by a retrotransposon events. Now, these footprints or the footprints of these events are evident that in the direct repeats that flank the pseudogenes, the repeats have facilitated in the insertion back into the genome. These pseudogenes are usually found a long way from the functional parental gene. Now, pseudogenes do not code for functional proteins and are not been transcribed. Now, the exact number of the pseudogenes in the human genome is actually unknown, and it's just been predicted, but it's not been known for sure how many are there together. Although the estimates using different criteria has identified something like 3000 regions which actually possibly represents processed pseudogenes. Now, the pattern that has actually emerged from the analysis of the human genome is that those sequences tend to give rise to more pseudogenes are shorter than average transcripts and are sequences that are actually involved nuclear regulations and translations, like for instance ribosomal proteins accounts for 67%, laminin receptor about 10% of the translations, elongation factors about 5%. The common theme among these sequences may actually be the increased level of transcription of these particular sequences. Now, the processed pseudogenes reported to date back several thousand years, and it actually falls into three categories: those that were actually complete copy of the mRNA transcribed from the functional genes, those that are only partial copy of the corresponding RNAs, and those that actually contain sequences in addition to those expected to be present in the RNA. Transcriptional regulation in the eukaryotes is a very complex process, often involving enhancers and the upstream binding sites for these regulatory elements. Now, these regions can actually cover thousands of base pairs. Now, intergenic DNA is actually been estimated to occupy about 63 to 75% of the total base pairs in the human genome. Now, the longest stretch of non-coding DNA, termed the gene desert, is actually present on the chromosome number 13. It is 3038, 416 base pair long. This was the big event of the last decade of the last century. The whole genome analysis has confirmed the early observations concerning gene density in the eukaryotes. Now, below is a table that we will be showing in the text. We will have some general statistics which has actually been obtained from the Human Genome Project. Now, modern insights into the C-value paradox. Now, the C-value paradox has traditionally been described into three different ways. More complex organisms do not always have larger genomes than simple ones. The quantity of the DNA does not seem to be related. The quantity of the DNA does not seem to be related to the number of the genes, for the amount of the DNA does not increase univocally with the complexity and the number of the hereditary characters. Now, what are the suggested mechanisms for the increase in the size of the genome? Now, that is actually a wonderful article which is published back in Nature. Now, any given genome seems to contain more DNA than that will be needed for the predicted gene numbers. One of the problems of the eukaryotic genetics is that higher organisms possess much more DNA in the genomes than they are likely to need in the genetic informations. Now, from this what people found that there is a poor correlation, again coming back to the same thing, that there is a very poor correlation between the DNA content and the organism's complexities. And this is in further revealed when people actually started doing the renaturation kinetics, and they found that the different regions of the DNA actually have to digest with the different kinetics. Now, the thing is that what exactly is that extra piece of the DNA that is actually been doing? Well, that extra piece of the DNA does not have a known function right now and is actually regarded as a junk DNA, but there is a fantastic project that actually been started in collaboration with NIH, and that is the Human ENCODE project. Actually, ENCODE stands for Encyclopedia of DNA Elements, ENCODE. Essentially, what the ENCODE does is that it actually catalogs the entire region of the DNA into the functional domains, functional elements, and tells that, okay, this is the enhancer element, this is the silencer sequences, this is the promoter elements, this is the coding regions, this is the non-coding regions. So, it is going to give a detailed map of the different regions of the DNA. Essentially, it is an anatomy of the entire DNA. When people started doing ENCODE DNA and then they brought about this junk DNA or the useless DNA that just increasing the volume without any functions, then they found that no, that is not right. In fact, even the non-coding DNA, even the DNA that actually doesn't code for the proteins, also codes for a large number of different microRNAs and other non-coding RNAs. That is an extremely important and extremely fantastic regulatory functions. So, the Human ENCODE project, which is a follow-up of the Human Genome project, sequenced the DNA that makes up the human genomes, and ENCODE project seeks to interpret this particular sequence, and it has been bound by a particular transcription factors. So, in the ENCODE project, what people found is that whereas in the genome actually transcription factors actually been bound, and they did found is that there is a heterogeneity in the DNA complexities. And it has been very important to know that this heterogeneity in the DNA complexities is because of the reason that different regions of the DNA has different sequence contents. And the C-value paradox is mostly in this line, that it is a way to understand that can you correlate the complexity of the DNA with this content of the DNA, and then we found that really there is not. So, ENCODE the DNA is a first way in the first project, is a mega project that actually tells beyond a reasonable doubt that look, the regions of the DNA which just adds to the volume does not have a function, still has a function, they are not essentially junk. They code for a lot of different regulatory RNAs, and this regulatory RNA is actually very crucial for the function of the organisms and the entire transcriptional networks and complexities and maintaining everything. Because if you remove those particular non-coding RNAs and the regulatory RNAs, then the entire transcription machinery shuts down, doesn't work properly. And therefore, ENCODE is an approach to counteract this particular C-value paradox and tell that look, now we know the function of this junk DNA, and they are not essentially junk, they are very vital DNA performing real important functions. Thank you.